We all know that blood is the red fluid that courses through our circulation and leaks out and clots when we cut ourselves. But what is it made of? Blood is a tissue and forms part of the circulatory system. It differs from most tissues of the body in that it is fluid, comprised of water, small molecules, and a regulated mix of cells and proteins, but no solid matrix. Water circulating in the blood accounts for about 20% of the extracellular water, the remainder being in the interstitial space surrounding the cells of tissues and organs. Blood communicates with the interstitial fluid, from which it is separated by the endothelial cells that form the walls of vascular capillaries. The endothelial cell essentially acts as a semi-permeable membrane, which allows rapid exchange of small molecules and water. Because they can readily exchange between blood and interstitial fluid, the concentration of small molecules is equal in the two compartments. For a bioactive molecule that easily permeates the capillary wall in this way, measuring its concentration in a blood sample gives a good idea of its concentration in the interstitial fluid around the cells it might affect. This is helpful when assessing the concentration dependence of its effects. In contrast, cells and proteins cannot usually cross the endothelial cell barrier, so the blood and interstitial fluid have distinct cell and protein compositions. The different components of blood can easily be separated by spinning a collection tube containing a blood sample at high speed in a centrifuge. The centrifugal force from continuous revolutions pushes the denser, heavier particles to the outer edges of the sample, resulting in distinct layers. Centrifugation separates blood cells from the liquid component. The red blood cells collect at the bottom of the tube and make up about 40 to 45% of the blood volume. This component is known as the hematocrit and is measured routinely in clinical blood tests. The liquid component is called plasma. It contains mainly water, ions, and proteins. A third thin layer, known as the buffy coat, forms between the red cells and plasma and contributes less than 1% of the volume of blood. It contains white blood cells and platelets. If the blood sample is left out on a bench before centrifugation, it forms a clot or a thrombus. When the sample is centrifuged, the clot is compressed at the bottom of the collection tube. The liquid above the clot is known as serum. The composition of serum is like plasma, except that it lacks protein clotting factors, which are used in forming the clot. The red colour comes from the red blood cells, sometimes called red corpuscles, although their scientific name is erythrocytes. These cells are around 7.5 microns in diameter and have a biconcave shape. Each erythrocyte is packed with the protein hemoglobin, shown here as a 3D model. The hemoglobin molecule contains 4 key structural motifs highlighted in green. Each of these motifs consists of a cyclic porphyrin ring known as a heme group. At the centre of the heme group is a high affinity binding site for iron in its ferrous state, which forms coordinate bonds. It is the iron in hemoglobin that gives it and blood its red colour. So, what are blood cells and where do they come from? The formation of blood cells is termed haematopoiesis, and it takes place in the bone marrow. It begins with a pluripotent hematopoietic stem cell. This name means that it has the potential to differentiate into any blood cell type. It first differentiates into a progenitor cell or starting cell for either of 2 cell lineages, the myeloid line or the lymphoid line. The lymphoid progenitor then becomes a lymphoblast before maturing into the fully formed lymphocyte. The myeloid progenitor has the choice of differentiating into one of three different cell types. It is the progenitor for erythrocytes. Alternatively, it can develop into a megakaryocyte. This is one of the largest cells in the bone marrow and contains multiple copies of DNA due to replication without cell division. As these cells grow, they extend projections into blood vessels that break off to become platelets. So, platelets are not really cells, but pieces of cells that lack a nucleus. The third cell type derived from the myeloid progenitor is a myeloblast, which can further differentiate into a basophil, a neutrophil, an eosinophil, or a monocyte. The first three of these are together known as polymorphonuclear cells, due to them containing granules and having multi-lobed nuclei. The four cell types derived from myeloblasts, along with lymphocytes, make up the white cells of the blood, also known as leukocytes. The pathway that differentiation follows is determined by a range of hematopoietic growth factors that control each step. These, in turn, are subject to regulation depending on the body's needs. The different cell types serve different functions in blood. Platelets are involved in blood clotting. Erythrocytes transport oxygen around the body, and the white cells are crucial for the body's immune response, protecting against infection, foreign invaders, and abnormal cells. The number of erythrocytes circulating in the blood is tightly regulated to maintain oxygen delivery to cells and tissues while preventing excessive blood viscosity. The need for new erythrocytes is sensed in the kidney as blood flows through it. What the kidney actually detects is a drop in the blood oxygen level, known as hypoxemia. Oxygen levels are sensed by specialised peritubular fibroblasts in the cortex and outer medulla of the kidneys. When oxygen is low, these cells release the hormone erythropoietin into the circulation. It acts in the bone marrow to stimulate the production of new red blood cells, known as erythropoiesis. It acts on the erythropoietin receptors on the surface of erythroid progenitor cells, which are in the pathway of myeloid cell differentiation into erythrocytes. The production of red blood cells can increase 6 to 8-fold when stimulated. This regulation is important for maintaining a healthy hematocrit and appropriate levels of oxygen in the blood and tissues. Plasma contains important proteins. The most abundant protein is albumin. It makes the largest contribution to the colloid osmotic pressure of blood. That is important because the colloid osmotic pressure draws water into blood through the capillary wall and retains it in the circulation. The protein has many ionizable groups, which help to buffer plasma pH. Albumin also binds many low molecular weight molecules and can therefore act as a carrier for poorly soluble small lipophilic molecules. This means that it can bind many drugs, lowering their free or active concentration in plasma. As different drugs can compete for binding sites on albumin, this property underlies important drug interactions. When two drugs that bind albumin extensively are administered together, the plasma concentration of each drug will be elevated compared to the concentration reached when the same dose of drug is given on its own. Another important protein is fibrinogen, which plays a key role in the clotting of blood. As with all proteins in plasma, it contributes to colloid osmotic pressure, but its contribution is relatively small. Another major group of proteins is the globulins. They are a broad class of proteins that serve many functions. These include transport, enzymatic activity, hormones, and immune functions. Transport functions include the binding of lipids by lipoproteins, which are present as high density, HDL, and low density, LDL, lipoprotein. These proteins are measured in blood tests as a marker of cardiovascular health. Transferrin is a globulin that transports iron around the body. The main enzymatic function of globulins is in the clotting cascade, which involves a sequence of events under the control of enzymatic clotting factors. A key example is thrombin, which cleaves fibrinogen into fibrin. Fibrin then polymerizes into a mesh that provides the structural framework for a clot. The hormone erythropoietin is a globulin, and as you have heard, it regulates haematopoiesis. As the name suggests, the immunoglobulins play a central role in the immune system, as they are the antibodies that neutralize the effects of pathogens. Together, the globulins contribute around 6 mm of mercury to colloid osmotic pressure, about 20% of the total.